To characterize the risk an emission of chemicals poses to the environment, both a dose or concentration, response, effect assessment, and an exposure assessment are performed. So the objective of the concentration effect assessment is to predict the concentration of the substance below which effects on the environment and people are not expected to occur. So this is this predicted no effect concentration. And in these discussions, the concept of no effect has a very dominant place, while the concept itself is rather vague. And different things have been proposed for no effect. And this talk will focus on the history and development and application of biology-based methods which propose the no effect concentration. The outline of the talk is to go through the history, to discuss and present applications in ecotoxicology, to discuss the notion of families of models, and of finally data and models. This entire story of biology-based models started in 1979, where Bas Koyman, here present at the time and starting in TNO, had to find answers to two simple concrete questions, which was basically how should we quantify the effects of chemical compounds on the reproduction of daphnids as revealed by standardized assays. And the second question, what harm does this small reduction in reproduction by toxic stress do? And the questions turned out to be related and much more fundamental and much less simple than they appeared at first sight. The answer to the first question led to the insight that reproduction cannot be studied by itself, that it is intimately linked with the full energy budget, which can be affected by chemical compounds in different direct and indirect ways, involving several timescales. The answer to the second question led to the notion that it concerns ecosystem dynamics and relates to properties of individuals. When DEB research started in 1979, existing models for population dynamics did not relate properties of individuals to the properties of the populations. They treated all individuals of a species identically. Chemical compounds, just like evolution at another time scale, change the properties of species, of individuals, and consequently have impacts on populations and on ecosystems. Differences between the individuals of the same species are key to evolution, as well as to complex links between levels of organization. And the individual unit is fundamental to biology. Since both questions had an environmental risk assessment context, the ability to make predictions for potential effects of toxicants was essential for situations which have not been tested experimentally. The standard empirical methods, so the LC50, are useless in this respect because they don't allow extrapolating beyond the range of observed conditions. And so at the time, this constrained the quest for methods which are based on first principles. It turned out that the knowledge about energy budgets of individuals was essential to answer the questions, and the way that chemical compounds affect budgets of individuals is vital for the evaluations of consequences at higher levels of organization. It appeared that many aspects of these energy budgets are not species-specific and apply to all species, microorganisms, plants, and animals. So it turns out that chemical compounds are one of many factors, temperature, parasites, food availability, that modify properties of individuals, and that there exists a unifying way to deal with such perturbations. Dynamic systems theory provided the proper context to capture how individuals cycle their way through life by changes in state variables, and how factors modify this by changes in individual-specific parameter values. And it shows that if you change particular parameter values, it works out in particular changes in growth and reproduction. And there is data and information which support this idea that there's a unifying way to deal with organisms, with processes, with modes of action of toxicants, and how they work out. So dynamic energy budget theory covers many things and has had many interesting applications, also in the medicine for tumor growth, in the wastewater treatment, with some very interesting work on biodegradation modules. But here I focus on ecotoxicology and applications in ecotoxicology. The applications in ecotoxicology remained on the agenda since the start in 1979, because that very first question at TNO with those daphnids was in an environmental risk assessment context. And the questions are very, very contemporary. All of the applications rest on the idea that effects relate to internal concentrations of compounds. So we have to consider availability, toxicokinetics, and transformation modules. And the DEB model specifies the parameters that can be potentially affected by the compound. The analysis of standardized bioassays, it's a basic result, but where the applications become very powerful is concentrations of toxicants vary in time, when the compounds interact with the physiology of the organism, when we have mixtures, or when we want to make steps from effects on individuals to that of populations, or other types of extrapolation. It's not about the model as something which is set in stone, and there is a link between the model complexity and the information we have and the type of data. Many empirical models, it's hard to read on this slide, but it starts with Lavoisier, so it's multiple regression of heat against mineral fluxes. We can go to Kleiber, 1932, respiration scales with body weight to the power 3/4. Monod, 1942, growth of bacterial populations. All of these models, which are used to fit empirical data, turn out to be special cases of DEB models or very good numerical approximations of them, meaning that today DEB theory is the best tested quantitative theory in biology and ecotoxicology. And the models, they can get larger or smaller in terms of parameters and state variables according to data complexity and assumptions. Perhaps more relevant here because we want to talk about sublethal effects and we talk about data, so you can think about endpoints. If you start to have an experiment where you measure multiple endpoints, you're measuring and growth and reproduction at the different ages of the organism, so over time, then you need a bioenergetic model, a DEB model coupled to a TK-TD module. So these are these modules for toxicokinetics and biotransformation maybe, etc. If you have a single endpoint, so you're only looking at growth, you still can use a DEB model. But you have more uncertainties as to what is the mode of action of the chemical. You can understand an effect on growth, but the reason why, and is it harder to understand, and the extrapolations are a bit harder. If you work with survival, you have this guts model, and if you have a stochastic death, then you're in this DEB-tox framework. You can have individual tolerance, you're in this critical body residue framework. Why would we want to work with families of models? What's the benefit? If in this case I can use this model, in this case I can use that model, isn't this whole family of model perhaps a rather academic thing? And in fact it's not, because if every single time I come with a new species, oh, now I'm working with bees, and bees are really special, and now I'm working with jellyfish, and jellyfish, they're, different because they have deuterostomians, or people find differences also depending on which discipline they're working on and the way they're doing their measurements, and then you can have very special models to take into account. And then suddenly you end up with a Christmas tree, and you start to have equations and parameters for every species, every geographic region, every compound. In DEB theory, for example, you have the aging module. So you have the standard DEB model, which was what Jan Bass presented. And then you say, now I have an aging module. Aging module comes with parameters and state variables. The energetics of the organism, so the food level, impacts the aging. But the parameters and the state variables of the aging module don't change what's happening with the dead model. So you're able to remove the aging module with no impact on your standard dead model. For example, if you work with particular fish species and all the eggs don't conveniently come out at once, the fish stores all of its reproductive material and then transforms some of it into ripe eggs, which is released at particular moments. So you might need a module to specify this, the dynamics of the spawning, how many eggs per spawn, what is the interval in between spawns. If you have a compound which is eliminating through reproduction, this is far from trivial because the change in the body burden of the animal can, be instantaneously decreased by a lot. If we now want to have a more applied project, where we say, okay, now we need to study or develop some sort of risk metric for produced water in the Barents Sea, to name a random example. How long is such a project going to be? Is it going to be 1 year? Is it going to be 3 years? We would like to have some results. We want a tool which is working. So we want to make use of what science, more fundamental basic science has developed. And then we can look and say, all right, how many testing programs, what are the testing programs the models have undergone? And depth theory has a 35-year testing program against a lot of experimental data in a lot of different disciplines. Is such a 35-year testing program going to happen again? There are many different types of data. So this is the third argument about families of models. So for example, you mentioned, Lars, that there were this cod experiment over 2 generations exposed to alkophenols. For example, if something like that could be analyzed with a dead model for sublethal effects, we'd still want to extract from that some sort of no-effect concentration. We want to extract for that parameters of an idealized toxicokinetic module, which is telling us something about the dynamics of internalized alkylphenol, which is giving us something on how maybe growth or development of that cod was affected, which is different than looking at hormonal effects. I mean, the time scales, etc. It's not the same thing. But for a population of cod, we need to come back to the growth, numbers of individuals, say recruitment. So by working with families of models, we're still able to extract comparable parameters, such as the no-effect concentration, while having very different types of data with different levels of information content. And it's possible to develop perhaps a scheme where we can say, well, this no-effect concentration and these parameters come from an experiment which was actually rather rich in information content. And this no-effect concentration, well, is coming from a test which was perhaps shorter than the time it would take for the toxicity of the organism to build up, so we're a little less certain in that value. But nonetheless, we're looking at no-effect concentrations, which are having the same interpretation. In some way, with respect to our topic, which is effects on organisms, one way to rank data could be thinking of static versus dynamic exposure and composition. And why standardized tests say exposure has to be constant. There's historical reasons for having to have constant exposure in a bioassay. That's not actually a necessity. So you can find in the literature pulsed experiments, for example. You have experiments with single versus multiple endpoints. Sometimes it would have been possible to get multiple endpoints, but only one endpoint was reported because the protocol said only report that. Then you have partial to full life cycle testing. There's a whole section. I was looking quickly some years back in the OECD, and they have these special fish tests with different, for guppies or zebrafish, which go for several generations. We could also use those by analyzing them with these type of models. Again, if points in time are reported. Then you have experiments with single versus mixture of compounds. The European Union is prioritizing and asking that we integrate more biology and more ecotoxicology into risk assessment. So we have to think, what does that mean? How do you integrate more biology and more ecotoxicology into risk assessment using what we have, being able to take into account more complex information from the scientific literature, and being ready for having feedback between the new experiments which are going to be done, and again being able to incorporate this into our assessments without changing everything. Is the structure of how we build our assessment tool, the underlying structure of it, is it ready to be incorporating new type of information? And I think the current system just isn't. It's not just that the EU is putting us in a strict system that we can't move anymore. The reason we can't move also anymore is because the actual way everything is built with these NOx and ECXs, and then there's no way we can have other type of data. We simply can't get NOx with different type of data. Possible. We have to only have the standardized assays. So we can think that when we build a tool, that while we don't solve all of the problems today, and while we still have a number of uncertainties, that we have the foundations that we can incorporate the data from future experiments, and these biology-based models are designed for that. There's a large database of data and depth model parameters. So I started my PhD in 2009, and that was when this started. There were very few species at the time, and I came with my zebrafish, and then Bas was at the time at the Vrije Universiteit and asked me what zebrafish data I had, and I came with all these data sets, and that then became a zebrafish entry. And ever since, the collection has grown, and the code and software has also been improving. Here we have a graph of entries as function of year, and today we're at 411 entries, and we're working on a formalized procedure to curate the entries, so entries can be submitted almost in a journal format and go through one of the curators. At the moment we're a board of 5 curators from 5 different countries. Here we have a pie chart, because we like pie charts, of the different groups which are represented in the collection. The collection is more or less complete for the large phyla, excluding sponges. Fish, mammals, and protostoma are the best represented groups, and chordates are complete at the order level. That DEB models can be used to estimate the no-effect concentration, that it has been worked out for standardized tests with this DebTox book, and it's also in the chapter of the OECD and ISO. But I also am saying that it can be done for more complex experiments. I would think that something which Chaling will talk tomorrow about, which is an important aspect of this workshop, and I think also one important aspect of these models, is that it is probably, as far as I can see, one of the more promising ways to be dealing with extrapolations. So the extrapolations, or let's say transferring knowledge from something I know to something I don't know. I think that it's always tricky to be looking in patterns. Perhaps we see patterns for the wrong reason. There's often a lot of noise in the signal, which can make it difficult to come to conclusions. Jan, you said that in the 90s the study of sensitivity of chemicals between species was more popular, that it grew less popular. It's coming back a little bit today. Looking at correlations between energy budget parameters, which have maybe a link with ecophysiology as well as taxonomy and sensitivity, is probably one of the more promising routes. But I leave that as open for discussion.